understanding about photosynthesis has a long history of tremendous research. Now we will see the structural organization of this photosynthetic system. As you know, there are two parts for the photosynthesis, the light reaction and dark reaction. Light reaction takes place in the grana and the dark reaction takes place in the stroma. In the grana, the light is trapped by a system of proteins and pigments. In the figure you can see the arrangement of these systems, the photosystems, the photosystem 1 and photosystem 2. These systems are transmembrane structures in the grana thylakoid, and various components included in it are the thylakoid proteins photosystem 2 and photosystem 1. The photosystem 2 is also called as P680, and there are some thylakoid proteins. One among them is the plastoquinone and the other is the plastocyanin. Both this plastoquinone and plastocyanin are mobile. They can move along the membrane from one photosystem to the other. So there are two photosystems, photosystem 2 or P680 and photosystem 1 P700, and there are two mobile units, plastoquinone and plastocyanin. Then there is a cytochrome complex. The cytochrome complex contains cytochrome B, cytochrome F, etc. And this organization is the basic structural unit or structural organization for the light reaction. Again, there is a transmembrane potential between the outside stroma and inside the lumen. Here we talk about the membrane of the grana thylakoid. Outside this grana thylakoid, there is the stroma. Everything takes place inside the chloroplast. In the chloroplast, there is a stroma matrix, and in the matrix, there is the grana thylakoids. Now we are talking about the membrane of this grana. Outside this membrane is the stroma. Inside the membrane is the lumen of the thylakoid or lumen of the grana. And these proteins, these structures, photosystems are transmembrane proteins on this membrane. So we can see the photosystem 2 here. Next, cytochrome complex is here. Then the photosystem 1 is here. There is a transmembrane potential between these membrane. There are ions outside the membrane and ions inside the membrane. These ions cannot move freely. A transmembrane potential is maintained. The most important thing is the flow of these ions. How this ion exchange, ion transfer takes place, or the potential difference is balanced is the most important aspect. Now we start with the light reaction. Photosystem 1 and photosystem 2 have reaction centers. Photosystem 2 has the reaction center P680 and photosystem 1 has the reaction center P700. When the photosystem 2 is excited with a photon, one electron is ejected out from the photosystem 2 and it is captured by the plastoquinone. This plastoquinone is movable. It moves from the photosystem 2 to the cytochrome complex. During this movement, it can capture one proton from outside. It catches one proton from outside, then when it approaches the cytochrome complex, it releases the proton. So a proton, H plus ion, is transferred from the outside stroma to the lumen of the grana. The electron reaches the cytochrome complex. From the cytochrome complex, the electron is transferred to the plastocyanin. Plastocyanin is also a mobile protein which moves to the photosystem 1. The photosystem 1 also can accept photons, get excited and release electrons. It is a continuous process. The electron released from the photosystem 1 is captured by another mobile protein complex known as the ferredoxin. The peculiarity of ferredoxin is that the Plastoquinone and plastocyanin are inner side of the membrane, but the ferredoxin is on the outer side. We have seen that when the plastoquinone move from photosystem 2 to cytochrome complex, it carry one hydrogen ion from outside to the inner lumen, and again the electron is carried by the plastocyanin to the photosystem 1. From the photosystem 1, electron is ejected to the ferredoxin on the outer surface of the granule. From there, it is transferred to the NADP reductase, where NADP is converted into NADPH2. Due to this accumulation of H plus ions in the lumen and depletion of hydrogen ions in the outside, there is a potential difference of protons. It is known as proton motive force. This proton motive force initiates a potential gradient along the membrane, and this flow of protons takes place through a specialized structure or specialized enzyme known as ATP synthase. So the protons, the H plus ions, move through the ATP synthase. As a result, ATP is synthesized to the stroma side. The energy flow of ATP synthesis takes place in the form of proton flow through the enzyme. Here we can see the structure of a ATP synthase. It is just like the system in mitochondria, the oxysomes. Here you can see the flow of protons, the electron flow takes place from the photosystems to the NADPH, and the flow of protons through the ATP synthase, resulting in the reduction of NADPH2 and synthesis of ATP. We have seen clearly the structures present on the membrane of the chloroplast and the mechanism of the synthesis of NADPH2 and ATP. So we can describe the light reaction. The first step in light reaction is the excitation of photosystem 2 with a photon. The electron jumps out of the photosystem 1. The plastoquinone, it passes through the cytochrome system, the plastocyanin, and to the photosystem 1. The photosystem 1 also get excited with the photon, and one electron is removed from the photosystem 1. Then there is a deficiency of one electron in the photosystem 1, and it is compensated with the electron coming from the photosystem 2. And that electron is transferred to the NADP reductase enzyme, which reduce the NADP to NADPH2. So the path is very clear. The photosystem 1 loses one electron, it is transferred to the NADP to form NADPH2. That gap is filled by the electron coming from the photosystem 2. Now there is a deficiency of electron in the photosystem 2. This deficiency of electron in the photosystem 2 is compensated by the electron coming from the photolysis of water. This is the actual mechanism of non-cyclic photophosphorylation. The cyclic is also very easy to understand, because the far red light or the light energy with higher wavelength is captured by the photosystem 1, and photosystem 2 is not activated here. From P700, the electron get excited and go to the ferredoxin. From ferredoxin, the electron goes to the plastoquinone. The plastoquinone carries this electron to the cytochrome complex. As a result, one proton carried into the lumen, and the electron is carried to plastocyanin, then comes back to the P700. This proton carried into the lumen passed through the ATP synthase and ATP synthesized. So there is no reduction of NADP or movement of electrons to the NADP reductase, but it goes back to the plastoquinone, cytochrome complex, plastocyanin, P700, come back. So it is known as the cyclic photophosphorylation. So we can say the difference between cyclic photophosphorylation and non-cyclic photophosphorylation. In cyclic photophosphorylation, P700 get excited and the electron is passed to the pyridoxine, plastoquinone, cytochrome complex, plastocyanin, and back to the P700. So the electron is transported back to the same chlorophyll molecule. But in the case of non-cyclic photophosphorylation, the electron excited from the photosystem 2 is transported to the photosystem 1, and the electron from the photosystem 1 is transferred to the NADP, where it gets reduced to NADPH2. And the deficiency formed by the ejection of electron is compensated by the electron coming from the photolysis water. In the case of non-cyclic photophosphorylation, there is a synthesis of NADPH2 molecule, but in the case of cyclic photophosphorylation, only ATP molecule is synthesized when the electron is passed from plastoquinone to the pyridoxine. One intake of one proton takes place. This proton, when comes out through the ATP synthase enzyme, one ATP synthesized. So there is no formation of NADPH2 in cyclic photophosphorylation. In the next class, we will see what is meant by photolysis, how the photolysis of water takes place, what is the mechanism of photolysis of water, and the completion of this light reaction. Thank you.